Hello and welcome to the Neuroinflammation and Neuroimmunity session. We have an exciting series of talks lined up in this session, starting with genetic discoveries in this pathway, all the way to possible therapeutic interventions by altering neuroimmune and inflammatory processes. My name is Badri Vardarajan, and I'm a bioinformatics scientist at Columbia University. And the following are my disclosures. Neuroimmune processes and neuroinflammation were initially considered as passive bystanders in Alzheimer's disease. They were later bumped up to be a mediator of neurodegeneration. However, recent studies, both genetic and mechanistic, have shown overwhelming evidence that neuroimmune processes are important players in the Alzheimer's disease pathogenic cascade, and they're now front and center in Alzheimer's disease research. Early studies that evaluated the brain samples from Alzheimer's disease and Parkinson's disease patients observed microgliosis in regions with high density of plaques and tangles. Microglial processes were also found in close proximity to the amyloid fibrils. Microglia are resident phagocytes that are ubiquitously distributed in the brain. They swallow debris and apoptotic cells and are involved in synapse remodeling. The microglia scan the brain for pathogens and cellular debris, and when they encounter neuronal death of protein aggregates, they initiate regenerative processes at the site. They then migrate to the lesion site and trigger an innate immune response. In Alzheimer's disease, microglia are able to bind to the soluble amyloid beta for clearance. Thus, one hypothesis for AD mechanism is that the cytokine levels due to neuroinflammation might downregulate the A-beta phagocytosis receptors, thus reducing the phagocytic activity of microglia. Microglia function, such as tissue surveillance and synaptic remodeling, are disrupted by A-beta accumulation. For example, in the figure, early A-beta accumulation and acute inflammation spurs microglia to action. They clear A-beta and attempt to restore tissue homeostasis. However, the triggering factors and aggravators promote sustained exposure and immune activation, which leads to chronic neuroinflammation, which then results in more microglial activation, and then the cycle continues. This persistent activation and exposure to cytokines is thought to lead to neuronal degeneration and ultimately neuronal death. Recent genetic studies point towards immune-related pathways. Large-scale sequencing and GWAS studies have identified several genes related to microglia and immune-related pathways. TREM2 was the most prominent finding from two large sequencing studies in 2012 and 13 in New England Journal of Medicine, as shown in the top panel. Several variants in TREM2 significantly increase risk of Alzheimer's disease by 2 to 4-fold, which is comparable to the risk conferred by having one copy of the ApoE4 allele. The most well-studied amongst the TREM2 variants is the missense variant R47H, encoding an arginine to histidine substitution, which was identified in individuals of European descent and in Icelandic populations. However, this mutation is absent in African American and Chinese populations. TREM2, by definition, is a triggering receptor expressed on myeloid cells too, and it's highly expressed in the microglia. It mediates the phagocytic clearance of neuronal debris. Other mutations in TREM2 have also been found, and some of these mutations, such as the R47H, alter the phagocytic functions of TREM2 in vitro. Moreover, the R47H missense mutation also impairs TREM2 maturation and alters the shedding by alpha secretase. Rare coding variants in two other genes, PLCG2 and ABI3, were identified in a large genome-wide scan of 80 patients and healthy elderly controls, as shown in the bottom left panel. PLCG2 is phospholipase C-gamma-2, which encodes an enzyme, PLC-gamma-2, that cleaves the membrane phospholipid PIP2, propagating a wide range of downstream signals. PLC-gamma-2 belongs to the same interaction network as TREM2 and may be directly involved in its signaling pathway. ABI3 belongs to the ABI family of proteins, which are cytoplasmic molecular adapters. Not much is known about ABI3 function in Alzheimer's disease, But ABI3 is expressed in immunoreactive microglia and might differentially regulate their migration to AD brains. Our first talk in the session will be given by Eduardo Marcora, who will describe how AD variants preferentially influence regulatory elements, genes, and pathways in myeloid cells. He'll focus on mining results from large genetic studies to infer biological mechanisms from disease-associated variants. Eduardo will describe how AD variants are enriched in regulatory elements such as enhancers of specific cell types such as microglia and myeloid cells, but not in other brain cell types such as in neurons. The next talk will be given by Phil Dieger, who will examine monocyte-derived transcriptomic and proteomic data in Alzheimer's disease brains. Monocytes are a key component of the innate immune response. They play a similar role as the microglia, that is removing debris and dead cells via phagocytosis. However, monocytes mobilize much quickly following inflammation and proliferate at the inflamed site to differentiate into macrophages. In Alzheimer's disease, circulating and infiltrating monocytes clear vascular A-beta and parenchymal A-beta deposits. Often, resident older microglia cells are less efficient than circulating monocytes in removing A-beta deposits in the brain. Thus, elucidating the role of monocytes is critical for the treatment and prevention of Alzheimer's disease. Phil's talk will focus on understanding the omics profiles of monocytes and their similarities with the frontal cortex. Todd Goldie will review the inflammatory and immune function in Alzheimer's disease through the lens of genetic risk factors. Todd will give a detailed overview of inflammation in Alzheimer's disease and use genetic findings in TREM2, PLCG2, and ABI3 as a guide to propose immune manipulations that could alleviate the disease phenotypes. Peter St. George Hislop will give the next talk that will describe the structure of CD33 and its implication for targeting studies. CD33 encodes a microglial surface receptor and negatively regulates protective microglial responses. It was identified in an early large genome-wide scan of Alzheimer's disease. The identified genetic variant in CD33 reduces A-beta phagocytosis, and the carriers of the variant show increased amyloid beta deposition in the brain. Therefore, understanding CD33 structure for putative targeting is promising for AD therapeutics. The last talk in the session will be given by Kate Monroe, who will discuss a novel therapeutic approach for Alzheimer's disease by improving microglial function using a TREM2 antibody. She'll describe ways to promote innate immune functions of microglia that confer neuroprotection. To this effect, she'll describe an agonistic antibody-based approach to enhance TREM2 activity, which in turn spurs good microglial processes. I think this is a novel and exciting approach to AD therapeutics. I hope you find this session informative in understanding the critical role of the immune system in Alzheimer's disease. I'd like to thank the National Institute of Health and Aging and the Alzheimer's Association for putting together this session. Thank you.